Good morning, everyone. Thanks to Janet and the Alliance for Regenerative Medicine for having us here for the Cell and Gene Investor Day. As Janet mentioned, I'm Gbola Amusa from Chardan. Our vision as a firm is to identify companies that can create real value for society and generate exceptionally high investment returns. And we think disruptive innovation is one of the key paths to that. And with that vision in mind, it's really a great honor to sit here with Kathy Hai, who's a true innovator in the field of gene therapy and also an accomplished hematologist. We'll hear a lot about, hopefully, how Spark has launched a number of, Luxturna, which is a number of firsts in the gene therapy field. But Kathy, just to start, well, first of all, thanks for joining me.
Thanks.
Could you tell us a little bit about the history of gene therapy, especially post-Luxturna, which, as I mentioned, is the first gene therapy for a genetic disease. It's the first AAV-based gene therapy approved, and also the first pharmacologic agent for a genetic disease. Sorry, for an inherited retinal disease.
Okay, well, thanks, and I'm happy to be here today. I'm getting over a cold, and so my voice is a little lower than it usually is. I want to state that up front. I feel a little bit about this question the way that my father talked to me about my original Latin teacher, and he told me when Miss Mitchell talks about life in ancient Rome, I want you to listen because I think she was there. So I haven't been in gene therapy quite that long, but a pretty long time. And I think, as most of the people in this room know, part of the hope of the Human Genome Project had been that we would, with all the data from the Human Genome Project, combined with the nascent science of gene therapy, we would be able to turn genes into medicines for people who really lacked good therapeutic options. And as all of you know, it took a little longer to do that than it did to complete the Human Genome Project. Human Genome Project finished up in about 2003. Human gene therapy clinical trials began at the same time in 1990. But as you know, it took considerably longer to develop the first approved products in the field of gene therapy. And if you look back at the history of it, I mean, through the first decade of clinical gene therapy in the 1990s, there was increasing experience with this therapeutic modality, and it was generally safe, but did not have very good track record for efficacy. And then at the end of that first decade, there were a couple of different high-profile adverse events. And that generally scared away most people who were investing time and effort and money in the field. And the field sort of contracted back to mostly academic medical centers and people who really believed that it was going to work. And through that second decade, through the 2000s, there were increasing publications of successful clinical results. One of the first was ADA-SCID, and that data set eventually became Strimvelis, but there were increasing numbers of positive reports. We had a report about early work with Feretagene, which became Luxturna, and there were reports of positive results in other larger diseases like hemophilia. So through that second decade of clinical work, a number of problems were identified and solved. And then in 2012, the licensing of Glybera by the European Medicines Agency, it became clear that there was, in fact, a regulatory pathway to approval. And I think that, to me, those were the sort of major milestones in the 20-year journey from the first clinical trials to the development of a product. I think it's always important to underscore to people that if you look at other classes of therapeutics like monoclonal antibodies or bone marrow transplantation, things take 1 to 2 decades in clinical development to come to fruition. And that's because, there are a lot of reasons for that, but one of them is that, no matter how many experiments you do in animal studies accurately predict profiles in humans about 70% of the time. So you have to get in there and do the clinical investigation. You will learn about problems that were not anticipated in preclinical studies, and you got to get in there and solve those. So that, I think, is, I think the time frame for the development of gene therapy is reasonable.
Right. So has gene therapy arrived then?
Well, I really think that it is. I think that if you look at the pace of the licensing of monoclonal antibodies, the first monoclonal antibody was licensed in 1986. The next one wasn't licensed until 1994. So the first one was OKT3. The next one was 8 years later, RiaPro, which we still use in the coronary care unit. And then after that, you see the pace quickening. And by 2000, 8 of the top 20 biotech drugs were monoclonal antibodies. I think we are at the beginning of exactly that time in gene therapy. First product licensed in 2012, next product 2016, 2017, 3 gene therapy products licensed. And, so I think we're in that upward inflection right now. I mean, as Janet mentioned, we see a couple of things that are likely to come to application this year that I think most people feel good about in terms of likelihood of success of licensing.
Great. So as head of R&D at Spark, I'm sure you talk to a lot of investors as well as industry stakeholders like the FDA. At this point, though, and moving forward, what are some of the most underappreciated themes in the gene therapy space?
Underappreciated themes. Well, I mean, I think we are right now at a place where if we exploit a number of pathways that are already reasonably well worked out, I think that they can lead to more products. I mean, I think that we've learned a lot about introducing AAV into the retina, into the liver, and we're getting more and more experience with introducing AAV into the central nervous system. And I think, there are 25 million Americans with single gene disorders and 7,000 different genes involved. So I think the possibilities are pretty extensive. So I don't know if that's an underappreciated theme. I think it's a clear theme.
Sure, great. And then let's talk about the regulator. Some would argue that it's a pretty good environment currently. A few years ago, I remember companies saying that there's a lot of knowledge that's ramping in the various fields, and so the FDA also needs to ramp its knowledge of cell and gene therapies. Has that happened, and are companies getting the guidance they need to move forward expeditiously?
Well, I served on the FDA advisory committee for cell tissue and gene therapy. The office has a different name now. It's the Office of Tissues and Advanced Therapies. But I was impressed in the 5 years that I served on their advisory committee that they've done a pretty good job of identifying and recruiting people with real expertise in virology and vectorology to come and work at the FDA as regulators. And so to me, my experience with the US FDA has been positive in that the reviewers we've worked with, I would characterize as people who are knowledgeable and resourceful. And when I say resourceful, what I mean is that if you bump up against an obstacle, it has not been my experience that they will then come to you and say, bring me the broomstick of the Wicked Witch of the West to solve this problem. They'll think about what's data that you can really get that could address the problem and make it so that we can all move forward. And I think back to our experience in our very first AAV gene therapy trial for hemophilia. We had done extensive preclinical work in the dog model of hemophilia, and we felt that we understood a lot about the biodistribution of the vector. And then when the first subject was infused in the liver, we found vector in the semen. And obviously, nobody's ready for germline gene therapy, and there was a lot of concern, and the FDA had to stop the study. But they worked with us in a very rigorous and data-driven fashion to get on the other side of that obstacle. And now, in all of the trials where AAV is delivered systemically, you will see initially vector DNA detectable in the semen, but it always goes away. It's a dose-dependent phenomenon. It always disappears. And, they worked with us to get that first trial back on track.
So to me, one of the remarkable features of the past year is that we had a couple ex vivo gene therapies approved on phase 2 data. Agilis has been invited to file on phase 2 data, to my understanding, and Avexis it's seemingly going to file on their phase 1/2 data, their first clinical trial, with perhaps some phase 3 patients to go with that. So I guess that begs an important question. What constitutes a registration study going forward? And what other potential best practices can companies use in clinical trial design to move expeditiously?
Well, I always think that if you're dealing with a disease state, like congenital blindness, where you really can manage to arrange a randomized controlled trial, nothing beats the convincing nature of a randomized controlled trial design. When you're dealing with diseases that are fatal or that lack any other therapeutic options, and your preliminary data suggests that you're really changing the course of the disease, then I think both regulators and sponsors are well advised to consider other types of trial designs. I mean, none of us like to see a randomized controlled trial design where some people get randomized to no treatment if we know that within a year or 2 years they'll be dead. So I think, again, my experience with the regulators has been that they tend to be data-driven, and if you can make your case that you can establish the safety and efficacy without a randomized controlled trial design, then they will listen to you.
Right. So I understand some frameworks are due, especially for hemophilia, from the regulator, and that'll certainly help. But what innovations, what else is changing that can help clinical trial speed, whether it's new endpoints, new trial designs, biomarkers? What should we look forward to from companies?
Well, I really do think that with increasing experience in a space, that all by itself, in my experience, will shorten clinical development timelines. So, certainly in our first trial of AAV directed to the liver, every time something new was discovered, we had to stop, we had to spend time talking with the regulators, we had to work out a pathway forward and so forth. I mean, now I think, people file INDs for AAV administered systemically, and it's a much more straightforward pathway. So I think that for both sponsors and regulators, as the level of knowledge in a particular route of administration, vector, combination increases, you get time savings based on that. And I think that that will certainly be the case going forward, for example, for retinal gene therapy. I mean, if you think about Luxturna, we began phase 1 in 2007, and the drug was approved in 2017. And that was on a fairly modest size program of 12 patients in phase 1/2 and 29 patients in phase 3. And it still took 10 years. Well, I don't think the next retinal gene therapy will take 10 years. So I think there's something that comes from just experience. And obviously, biomarkers are key. I can't sit here and name for you which ones are likely to unlock timelines, but I think that increasing experience in the space is likely to yield some time savings there.
Great. So Kathy, I know you're not a commercial representative of Spark or an operations specialist, and it's early days for the Luxturna launch. But could you talk a little bit about your perspectives on how the launch will be different versus, let's say, traditional medicines in terms of what the uptake will look like or anything that might differ? And also, comment a bit on the role of your R&D organization in sort of helping the launch go well.
Well, there are a lot of questions there, Bo. But let me try to start from the part that I know perhaps the best. So, in contrast to some other AAV applications, which are straightforward, for hemophilia, for example, we infuse AAV intravenously, so that can be done in most hemophilia treatment centers. For Luxturna, as you know, this is a subretinal injection. So one of the decisions that we made early on is that, at least initially, the drug would be made available in a fairly restricted number of treatment centers. And so one of the areas of preparation was that we had to work with the pharmacies at these treatment centers to teach, management of the AAV vector in their pharmacy. And we also held a series of surgical workshops with the surgeons at the treatment centers to make sure that the learnings from the phase 3 study by our 2 principal investigators were transmitted to get the most uniform and best surgical delivery of the vector. So we had all of that work to do. In terms of direct involvement of R&D in the launch going forward, so first of all, as part of our approval, we did commit to 15 years of follow-up of all of the patients in the clinical development program. So we see those people once a year, and that is allowing us to collect data on the durability of the effect. Because we have a pay-for-performance aspect of our pricing and reimbursement, we also had to work with our colleagues to make sure that we introduced in a standardized way the metric that would be followed for the pay-for-performance for payers who agree to that. So it's a full-field light sensitivity testing. It can be done in the offices of most inherited retinal disease specialists, but we wanted to make sure it was done in a standardized way. We obviously are doing a safety registry on all patients who receive Luxturna for the first 5 years, so we're participating in that. And one of the areas where I think R&D will be most useful going forward is that a requirement for getting the drug is that a patient has biallelic mutations in the gene, RPE65. You have to have that. If you don't have that, the drug won't work. And so some people get genotyped, and they have what's called a variation of unknown significance. So it's not a mutation that has been previously described as causing the disease. Well, as part of our clinical development program, we developed a potency assay for RPE65. So we can take any mutation that's unknown, put it into that assay, and tell you whether or not that RPE65 has activity. So we have a lot of interfaces with the group. So, the other area where Spark put a lot of effort was into the area of pricing and reimbursement. And because this is a high-value, high-cost, one-time therapy, we thought about alternative delivery models because we want to make sure, we spent 10 years developing this, we want to make sure that anybody who needs it can get it. So we came up with 3 different potential approaches. One is, I mentioned that for payers who agree, we have committed to this pay-for-performance potential for a rebate, and that's based on improvement in full-field light sensitivity early on, 30 to 90 days afterwards, and also 30 months after vector injection. We have also developed an alternative to the buy and bill. So this Luxturna is like other specialty pharmaceuticals, and it can be purchased by the medical center, and then they would typically bill the payers, and there would be a markup. We have, for payers who agree to participate in this, set up a system where the payer directly pays, and so the medical center does not have any financial outlay, and the payer doesn't get the markup. And then third, we have applied to CMS for a demonstration project that would, if they agree, allow us to explore an annuity type payment for individuals covered by government payers.
Thank you for the perspectives on pricing. I'm used to hearing no one knows where pricing will go when I ask the companies. Actually, it is a sort of uncertain environment going forward for gene therapy pricing. We know that rare disease products can cost hundreds of thousands of dollars, and obviously if gene therapy is durable for 5 years, 10 years or more, it might justify very high upfront prices. So could you at least tell us the state of the art in terms of durability for what we've seen in AAV, whether in animal models or in humans, or actually both?
Yeah, no, that's a very important question. And, you know, this whole issue around pricing, I have to say, is so much more straightforward when you talk about disease entities that already have a treatment. So everyone in the room knows what people with hemophilia pay for clotting factor in a year. And in the United States, not counting if you have an inhibitor, if you just have regular hemophilia, you may be paying anywhere from $250,000 to $400,000 a year for clotting factor. Well, so what's the data that we have so far for AAV treating hemophilia? The most long-standing data that we have is from a trial that was sponsored by the University College London and St. Jude Children's Research Hospital. We actually collaborated on that when I was still at Children's Hospital. And the first patients were treated in March of 2010, and they're still expressing. So that's 8 years later. So those were levels that are, you know, they averaged around 5% of normal. And obviously, we wanted to get higher than that. So the hemophilia B trial that Spark sponsored that started in December of 2015. So we now have on those patients with average levels of around 35% factor IX, data out to about two and a third years with no, I lost my microphone here, with no evidence that it's diminishing. Trying to figure out if I need to move this. So in humans, data out to 8 years in the liver, and more, you know, all the data that's coming along behind that seems to back it up. In animal models, we have evidence out to about a decade. It's hard to get more than about 10 years of data in a dog model, right? Because they don't live longer than that. So, you know, I am confident that we're going to get long-term expression. Now, if you want to ask me, is it going to be 20 years or 30 years? Okay, that I can't answer with data.
Yeah. I think it's reassuring that some of the cost-effectiveness agencies are assuming durability. But one of the things that's evolving, especially in the U.S., is consortia that are forming, such as at the Margolis Center at Duke, to sort of bring together PBMs, companies, policymakers, et cetera. Do you foresee any of those initiatives playing out in the next 5 years, or do you think the U.S. market will be sort of status quo? How might the U.S. change? And it's okay to say we don't know, because that's usually the answer I get.
Okay, well, I have to say, honestly, I don't know the answer to that.
Okay. So let's talk about Novartis, which, as everyone in the room knows, is much in the news in terms of being a consolidator in AAV-based gene therapy. I think they found 8 to 9 billion reasons to go after Avexis. It's interesting that they partnered with Spark for ex-US Luxturna at what many argue are pretty good economics to Spark. Could you talk about the logic of having a big biopharma partner or collaborator to help launch a product like Luxturna outside the U.S.?
Yes. So what I would say about that, as you can tell from what I said earlier, we are really interested in pricing, reimbursement, and access for gene therapy. And we put considerable effort into the alternative delivery models that we came up with. On the other hand, I think we were trying to be practical about the likelihood that we could, in a very timely way, deal with health technology assessors across all the countries in Europe and through other rest of world where we would like to make sure people have access to Luxturna. And so we began to consider whether it might make sense to seek a partner. Novartis, as you know, has retinal products. They have front of the eye and back of the eye products in 100 different countries. And so in many ways, they seemed like a very desirable partner. So I think that by partnering with Luxturna, we can continue to invest more effort in developing new products. We have committed to commercializing Luxturna in the United States. We believe that we are very well qualified to make the case to payers about the advantages of gene therapy as a therapeutic, but we are also practical about our bandwidth at this point.
Great. So let's talk manufacturing. We know how important R&D is to setting the tempo of a product in terms of clinical trials progression, but it seems like manufacturing is as well. In AAV-based gene therapy or gene therapy in general, on a scale of 1 to 10, how would you frame the importance of manufacturing for clinical progression?
Well, I mean, look, from the earliest days that I have worked in gene therapy, which is now well over 2 decades, there's nothing, I mean, you need good ideas, you need good targets, you need to know how to execute on clinical trials, but if you don't control the manufacturing, you don't control your fate. And so to me, that's always been a cornerstone of all efforts for product development in gene therapy. And I was very fortunate in the early work that I did to work with a terrific biotech company on the West Coast in the 1990s, Avogen. And when all of the gene therapy companies went away, we actually were fortunate to be able to retrieve a number of the manufacturing people from Avogen and get them to come work at the Children's Hospital of Philadelphia. And subsequently, they went with us to Spark. When I look back on that, and I think that all I had to do to get those people was convince them that living in Philadelphia was a lot like living in San Francisco. I'm always amazed that I was able to get them to come, but we did get them to come, and they really formed the cornerstone of our manufacturing group at the Children's Hospital of Philadelphia, and then subsequently at Spark. But what I've seen over the years in gene therapy is that it's really interesting. It's almost like clinical development and manufacturing go like this. So if you get good clinical results, it allows you to find the funding to invest more money in manufacturing. And then when you have better results from manufacturing, you can exploit that to do even more things clinically. And what I've seen is that good clinical results drive investments in manufacturing. And when I think back on the amount of vector that we could make 20 years ago and what we can make now, it's really been an extraordinary odyssey. But I think that I have confidence good clinical results really push investment in and progress in manufacturing. And so, without any question, manufacturing of AAV is a very complex undertaking, and reliable, consistent manufacturing continues to be an important goal for the industry. But I believe that when I look at the progress that's been made over 20 years, I'm not worried about whether that will eventually limit what we can do.
Right. So I mean, we all know it's fairly important, but I mean, unless we get the keys to a company's labs, which none of us, well, actually, I assume none of us are going to get. I mean, how do we, from the outside, assess a company? I mean, what should we be looking for? What are some of, we hear about consistency, batch to batch, et cetera, but are there any tangible signs that we can use to say this company's pretty good and this one maybe not so good?
Well, I think that's a harder question to answer because, you know, as you know, what seems to happen in the industry is that people are fairly clear about their clinical results. They're published. You can read about them. They're presented at the advisory committee and so forth, whereas everything about manufacturing tends to be much more subterranean. And I didn't make that rule, but it seems to be the rule. So from your perspective, how can you tell? I think that's, you know, a little trickier. You know, look for pauses in clinical development that don't have another explanation.
Okay, great. Let's talk systemic toxicity. Recent concerns were raised in academic literature. What are your perspectives on toxicity from high doses of AAV-based gene therapy?
Well, you know, this is an interesting question, and I think it's related to your previous question. And, you know, I think we're all familiar with the results from the spinal muscular atrophy trials. And they're giving very high doses of AAV to infants, 2 times 10 to the 14th vector genomes per kilogram. And the safety track record has been excellent. They had a little bit of transaminase elevation. No, not a little bit. They had transaminase elevation early on and put all the patients after that on prednisone. But other than that, and I heard a presentation about this last week, the data look great. So you can give very high doses to people and not have a problem. Other sponsors, and one in, pigs and non-human primates, and another in a clinical trial for DMD, have had much more marked toxicity at the same dose. And I think what that tells you is that all AAV vectors are not created equal. And so the things you would want to know, but that you won't be able to find out, I suspect, are very basic things like, oh, if I run this out on a silver stained gel, does it look clean, like there are just 3 bands? Or does it look like a black smear because there's a lot of other material in there? I think when you're trying to make very high doses of AAV, you have more to worry about in terms of product-related impurities. And people have differing levels of success at making a high-quality product. So that would be some of what I had to say about that. I would not conclude that nobody can make AAV safely to give it doses of 2E14 per kilo.
Right. So it sounds like manufacturing matters.
It matters.
I like saying obvious things.
So do I, Bullet.
So let's talk about the future. What innovations do you think are upcoming that could affect regulatory success and disruptively innovating in gene therapy?
Well, I mean, I feel a little bad that we've spent the whole time talking about AAV, but I'm sorry, I can't help it. You know, there are clearly some problems that remain to be solved with AAV. We can't package more than about 5 kilobases of DNA. We'd like to solve that problem. There are some people who have some clever ideas. Let's see how they pan out. If we want to deliver AAV systemically, we are limited by the fact that some people have pre-existing antibodies to AAV, and it's difficult to get past those. And so the field is working on ways to overcome those or to circumvent them. And then, of course, we would love to have a situation where we could repetitively administer AAV. For example, in hemophilia. Let's say that after 15 years, the effect does fade. Well, you'd like to be able to give it again. When we give it now, after the person gets it, they get high-titer antibodies to AAV. At Spark, we're working on a strategy that we're pursuing with Selecta Biosciences to block the formation of antibodies to AAV after the vector is given. And then you would presumably be able to give it again. So I think there are multiple lines of research that should expand the capacity of AAV to address serious diseases.
Great. I think we have about 5 minutes left, so I'll ask one more question and then open it up to the room. So from your perspective as head of R&D at Spark, and I kind of asked this about the industry, but now focus on Spark. What are people most missing about the Spark story in terms of especially sources for upside going forward?
Well, okay. I think one thing let me put it this way, one thing I'm very proud of about Spark R&D, we have 4 programs in clinical development. Well, Luxturna is already out of clinical development, but of the 4 programs that we have taken forward into the clinic, 3 of those have earned a breakthrough therapy designation from the FDA. And breakthrough therapy designation means that based on the clinical data you've developed in your clinical development program, the judgment of the regulators is that this represents an improvement over currently available therapeutics. So I think that's a pretty good track record. And I think it means that we're good at picking targets and we're good at executing.
So you must interact with the FDA a lot. What are recent learnings if you've met with them in recent times?
I find them consistent over a long period of time. Many people in the audience may know that the longtime head of the Office of Gene Therapy within the Office of Cell Tissue and Gene Therapy was the person that we recruited at Spark to be our head of regulatory. So again, I found them over a long period of time, data-driven, scientifically rigorous. I think if you can make your case to them, they're reasonable.
Great. Any questions in the room, please?
So you're looking at with regard to the third alternative you presented on pricing, are you looking at an assumable mortgage to account for changes in healthcare plans?
Not that I know of.
We have a few minutes.
Kathy? Kathy, you talked about I'm over here. You mentioned that you guys have really been forward-leaning in trying to come up with other kind of reimbursement models, both for private payers and for public payers, including the idea of kind of an annuity payment over time. Could you talk a little bit more about that? Maybe specifically what kind of reception you're getting to those alternative payment models, and in particular what the barriers are, if they've been identified, to an annuity model on the public payer side of things.
Well, I'm not an expert on this, and I do think that one of the barriers are the government price reporting requirements. And so that is why we've had to apply to CMS to consider this annuity model, because it will take some cooperation from them in order for us to explore it. I don't think I have much more to say about that.
Then one final question. Well, thank you, everyone, and a special thanks to Dr. Kathy High.
Thank you.